Bismillahirrahmanirrahim. Now, this is another video on the topic of pectinase, which is a new component of the 5090. But most of these questions are from 0610 because they are about the enzyme pectinase, which has been added to the syllabus. Pectinase is an enzyme which is used to extract juice from fruits such as apples. Figure 5.1 shows the volume of juice extracted from two equally sized samples of crushed apples, sized samples of crushed apple, not same mass, over 16 minutes. Sample A contained pectinase and sample B contained the same volume of water. So the pectinase was added as a liquid, so the same volume of water was added so as to compare whether pectinase did work or just the water worked as well. So we have on this graph, now I want you to look at the graph. This is time in minutes, and this is up to 16 minutes, and then this is the volume of fruit juice. So with pectinase, you see the volume has gone up to 120 centimeter cube here. And please remember to write the units. So 120 centimeter cube. And in this situation with water, it hasn't gone that much. It is somewhere just a little above 80. So this is about 82 cm cube. Now coming to part A of the question, how much juice had been extracted from the apples in sample A after 2 minutes? Now let's look at the graph and sample A 2 minutes. So you got to check here sample A and you have to be checking how much juice has been extracted from the apple in sample A after 2 minutes. So you have to see we have got minutes here. So 2 minutes sample A is going to be how much? Now this is about 38 cm cube. So the cm cube is written here. So just writing 38 here will get you your 1 mark. Then it says how much longer does it take for sample B to produce this amount of juice? Show your working. How much 38 cm cube, how long did it take? So you extend the line, the green line that I've just shown you. So you extend this line and then you see how much time did it take. So it took 6 minutes. Previously it took 2 minutes. So 6 minus 2 would be 4 minutes because what is it asking? 6 minus 2 is 4 minutes. So how much longer does it take for sample B to produce this amount of juice? So how much longer would it take? It took an extra 4 minutes. From 2 it has gone up to 6 minutes. So how much longer did it take for sample B to produce this amount of juice? So show your working. So this would be 4 minutes. Then state two advantages to the food industry of using pectinase in juice extraction. Now that's a question, why do we use pectinase in juice extraction? Basically we use it for what? More juice extracted, or you can say a bigger yield, faster extraction, or you can say it takes less time. And of course, if you have more juice, then you sell more of that, so more profit. So that is the why you use it in the food industry. So how do you write this? I mean, more yield, more juice extracted, faster extraction, it takes less time, and more profit, or you make more money. Then coming to B1, 5B1 of this question, yeast can be added to the apple juice uh, to make cider by anaerobic respiration. Define the term anaerobic respiration. Basically, what is anaerobic respiration? You break down nutrient molecules to release energy. That is the definition of respiration. Breakdown of nutrient molecules to release energy without using oxygen. So basically, like in human beings, glucose is respired to release energy. Similarly, in plants also, glucose is respired to release energy. Here in yeast, it could be any nutrient, could be glucose, could be also using any other molecule. So breaks down nutrient molecules to release energy without using oxygen because it's anaerobic. And please remember there were 2 marks, so you have to give me something like 2 points for it. So be careful when you are answering these questions, is that you need to give me more points looking at the mark schemes. As well as making alcohol, the anaerobic respiration of yeast can be used to make other useful products. Yeah, well, we can make bread. We can make biofuel. So these are the two that you could have written for this one. Yeast can respire aerobically. State two ways aerobic respiration differs from anaerobic respiration. That's very basic. One uses oxygen, the other does not use oxygen, releases more energy, produces water, does not produce alcohol. So you can always do this also. You can make a line on it, or you can do the comparative statement in the line. It's up to you how you can do this, but some of you feel very scared of doing this. And I said, well, there's nowhere it says that you can't draw columns on the lines on the. You see, everything, whatever is not allowed is written on the first page. That you do not use the ink eraser and all that. That's written on the first page. So if the most in such instructions is given on the first page, well, then don't assume those things. Be independent and have your own thought, because drawing a line and giving a comparative table would definitely, if you've written the correct things, well, you would get the 2 marks which are specified in this part of the question. Another question on pectinase. Pectinase is an enzyme that is used in the production of apple juice. A student investigated how pH affected the volume of apple juice produced when using pectinase. Basically, it's an enzyme question. The student chopped an apple into small pieces. The pieces of apple were put into solutions with different pH values. Pectinase was added to each solution. After 2 hours, the mixture was filtered and the volume of apple juice obtained was recorded. Table 6.1 shows the results. So here we have the results. So at pH 1, we had 23.2 cm cube of the apple juice. At pH 2, 24.2. At 3, it was 23.5. Then at 4, it was 25.7. At 5 pH, it was 27.6. At 6 pH, it was 27.4. At 7 pH, 24. And at 8 pH, 22. Now let's look at it. Where is the largest figure? The largest figure, as you can all see, is 27.6. You see it's increasing, 23.2, 24, then 23.4. There's a slight drop here. Then it's 25.7. This one looks a little anomalous. And then 25.7. And then on pH 5, it is 27.6. And then at 7, it has started to decrease. So if you look at the pH graph, it's something like this. So it would be the maximum value would be this one. So here, this pH is around about 5, and then it is starting to decrease. You see here it was starting to increase, and then here we see the optimum, and then so this is how you're going to figure this out. You must remember the pH graph whenever you're thinking of this. There's a very typical pH graph and temperature graph of enzymes. So after you've got this whole thing. You see, after 2 hours, the mixture was filtered. So we let this whole thing remain, the apples and the pH, the full solution of the pH, and of course the pectinase in it. And state the pH. Now let's look at state the pH at which pectinase is most active. Now that is 5.0. And it says give a reason for your answer. This is the pH where the largest volume of apple juice is produced. Why do you say 5? Why did I say 5? Because the largest volume, because basically what should pectinase do? It should break down the cell wall and release more of the juice from the cells. So 5.0 would be the optimum, but the reason for your answer has to be correct. That is what you have to be very specific about. 5.0, this is the pH where the largest volume of apple juice is produced. State one factor other than pH that would affect the activity of pectinase. Yeah, the temperature, concentration of the enzyme, or surface area of the apple. So you have to think of the practical when you were doing it. So all enzymes are temperature sensitive. They would be affected by temperature. If the temperature is less than the optimum, slow. If it's more than the optimum, denature the enzyme. Or you could have talked about the volume or the amount of enzyme, or you could have talked about the surface area of the apple. You see, you look in the question, and there's something which is they're telling in the question. The student chopped an apple into small pieces. Now, chopped an apple into small pieces. I mean, this is very, very non-scientific. How many pieces? In one there were 10 pieces, in the other there were 20 pieces. So again, this is again the thing which was something in the question which you had to come up with. So state one factor other than pH that would affect the activity of pectinase. So temperature, or you could have said the concentration of enzyme. If there was more pectinase or less pectinase, well, you remember if there is more pectinase means that 100 active sites. If there's less pectinase means 80 active sites. Or we could talk of the surface area of the apple because that was also not standardized, that it would all be chopped in a blender or something like that, which would standardize, or we'd have the blender on for maybe 30 seconds. So that would make it a little more uniform. Then comes to the B part of the question. Pectinase is an enzyme. The box on the left shows the beginning of a sentence. The boxes on the right show some endings of sentences. Draw three lines from the word enzyme to make three sentences. Now let's look at this. Enzymes are living organisms. Enzymes are proteins. Enzymes can only be used once. Enzymes have a complementary shape to their substrate. Enzymes increase the rate of chemical reactions. Enzymes in the stomach are most active at pH 8. Well, you know which ones are correct. So basically, it was very simple. Enzymes are proteins. That was correct. Enzymes have a complementary shape to their substrate, and enzymes increase the rate of reaction. Now, please remember it said draw three lines. So if you draw five lines, well, you actually negate yourself. You won't get any marks. This was the 3 marks, and if you drew another line here, like for instance, somebody drew this line, well, this was wrong. So 4, you'd get 3 minus 1 because it said draw three lines. Now you drawing another fourth one or another fifth one. Don't do that. If you don't know it, think about the answer. You know enzymes are not living organisms. Enzymes are found inside living organisms. Yes, you could have said that. You know enzymes cannot be used once. This one was wrong. Why was it wrong? Enzymes are used again and again. They remain unchanged, so they can be used again and again. And then enzymes in the stomach are most active at pH 8. No, it's the highest. It's very acidic, the pH of the stomach. It's nearly 3. Then it says state one use of enzyme in biotechnology other than fruit juice production. So we use it in biological washing powders. So biological washing powders will break down the stains of blood or protein or some sort of food thing which is oily, like maybe you dropped some mayonnaise on your clothes, so that would remove that. Then coming to the part D of the question, it says many types of enzymes are involved in digestion. State the names of two digestive enzymes in the human alimentary canal. Now that was very easy. I'm sure you could have done any of them. State the names. So you could have said amylase, you could have said lipase, you could have said a protease or a pepsin, because we have two proteases in the human alimentary canal, the pepsin in the stomach and the trypsin in the small intestine. So state the names of two digestive enzymes in the human alimentary canal. So any two. But please remember, if you wrote three and one was wrong, then I'm sorry, you're not going to get the 2 marks, because then there's a negative one. Plus one, minus one. You write one correct and one wrong, that becomes plus one, minus one. So you've written three. And of course, in these three, all are correct. But if you wrote me something else which was incorrect, then you would get a minus one. It's only a minus one when you negate yourself. When you're not sure, you've written, say the answer was xylem, and you've written xylem and you've written phloem. So xylem is the correct answer, but you've written phloem as well. So then you get a plus one and a minus one because you're not sure what is the answer, so you've written both of them. So this is the way you must understand, is when you write your answers, you need to be very you need to think about it before you write the answer. Now coming to another question on the pectinase. Figure 2.1 is a photograph of the cut surface of half an apple. Magnification is 1.2 times. Now it says make a large drawing of the apple shown in figure 2.1. So you just do a drawing of it. I'll try to do a drawing of it. It might not be very exact. Now coming to another ATP paper, which is on pectinase as well. So I'm going to be talking about that. Figure 2.1 is a photograph of the cut surface of half an apple. Measure the length of it, it's 10 centimeters. So you always answer in millimeter. Please remember that. Magnification is 1.2 times. Now, when you go it, you have to make a drawing of it. I've done a little drawing of it, not a very nice one. And there were 4 marks for this. And what are the marks for? So this outline, single, clear, continuous line, no shading. Size occupies at least half the space provided, so it has to be a large diagram. Then the calyx and the stalk, which has the thickness. Calyx are the sepals here, which you can see here. And 2 of the pips in it, which you can see here. So these are the seeds here in it. So you got your 4 out of 4 for this. Then it says measure the length of XY, which is 100 millimeter or 10 centimeter. They gave you the millimeter part here. Calculate the actual length. So you had to divide 100 by 1.2 and the nearest whole number. It says the nearest whole number was 83 millimeters, the actual length. Apples can be used to make juice. The enzyme pectinase can increase the volume of juice that is released from each apple. A scientist carried out an investigation to find out which concentration of the pectinase solution gives the maximum volume of juice. A similar question in the previous one. The results from this investigation are shown in table 2.1. So the percentage concentration of was 0.0%, 2.5%, 5%, 7.5%, and 10.0%. Volume of apple juice corrected, we did 3 trials and then this is the average. So you can see the largest volume was 23.7, and as we've increased the pectinase solution, but then as we've increased it from 7.5 to 10, then it has not increased. This is in fact decreased here. So identify the variable that has changed by the scientist in this investigation. This is always the first column. Independent variable is always on the first column, and this is always plotted on the x-axis. So the independent variable which that was changed was the percentage concentration of pectinase. Identify the variable that was changed. So this is what was changed, percentage concentration of the pectinase solution. Then part 3, the scientists decided not to include the trial 3 result for the 2.5% pectinase solution when they calculated the average. Explain why. Now, when you look at trial 3 for 2.5%, let's look at trial 3, 2.5%. Trial 3, this was 2. This was 11, and then it was 10, and then it was 2. So this looks like an anomalous reading. Anomaly means it's a mistake. It doesn't follow the trend. 11 and 10, it could be a 9 or could have been a 12, but 2 is very little as compared to. So these are called anomalous readings or anomaly, or you can say they are errors. So you could have said an error because the alternative words are allowed in this. The word anomaly, everybody should be very familiar with this word. We call it an anomalous reading. Then part 3 of the question, it says describe how to test a sample of apple juice to show the presence of reducing sugars. Give the result of a positive test. And there's 3 marks. So number 1 is add Benedict's reagent or Benedict solution, and then you heat in a water bath at any temperature between 80 and 100 degrees Celsius, and a positive result means that it's going to be brick red because Benedict's is blue in color. So the color will from blue go to brick red. Even any other colors, if you wrote, it was fine. If you said orange, even that was fine. If you said green, even that was fine. You see, when you're writing, you have to sometimes, just a careless mistake can cost you quite a few marks at the end of the day if you're making careless mistakes in 2 or 3 questions. So add Benedict solution 1 mark, heat in a water bath any temperature between 80 and 100 degrees was fine. Then a positive result means that the color change you're giving me, that Benedict solution is blue in color, so the color change will be from blue to brick red. The color change is, if it remains blue, it means no reducing sugar present. Doesn't mean no sugar present, because please remember they are non-reducing sugars as well. And the other sugars, what you have to understand, they're reducing and non-reducing sugars. So if you say no sugar, that's wrong. Benedict's test only is for reducing sugars. It is not for non-reducing sugars. So Benedict's is blue in color because it's got copper in it. And the copper actually takes part in the reaction, and that gives you the red copper precipitate. Then coming to the part C of the question, starch in apple juice makes the juice appear cloudy. The enzymes amylase can be used to remove the starch. This makes the apple juice clear. So starch makes the juice cloudy, but the enzyme amylase can be used to remove the starch. So it will break down the starch to maltose. This makes the apple juice clear. Amylase catalyzes the breakdown of starch. Plan an investigation to determine the optimum temperature for amylase activity in a sample of apple juice. So how would you plan to find out the optimum temperature for amylase activity? So if we're going to be investigating temperature, now this becomes the independent variable. So we're going to change the temperature. We're going to use different temperatures and study how much. So the independent variable is the temperature. So you should at least specify 3 temperatures. Then we're going to see the how much of the cloudiness of the juice is going to go away, because you said the starch, it said in the question that the starch makes the juice appear cloudy. So then the cloudiness should go away, it should become clear. Then how do you maintain the temperatures? Use a water bath, and then you measure at regular the samples at regular times. Then you use an iodine solution to test if any starch is present. What are you going to keep standards? The time, the pH must be kept the same, the volume of apple juice, the concentration of apple juice, the type of apple juice, the volume of the enzyme solution, and the volume of the iodine solution. Then you can always write, repeat the experiment at least twice or at least 3 times. And then, of course, you will give me one safety procedure is that when cutting the apples, be careful. Turn the scalpel away from you. The sharp side should be away from you so that you don't cut yourself. Now, as you can see in these planning and investigations, you have to give me quite a few points to be able to get these marks. So there's a very standard procedure by which we do these questions. Whenever it is asked for you to change, which is the independent variable, so you say at least 3 temperatures or at least 3 pH or whatever you have to change. Then how will you check the result? The result is always the dependent variable. So you will check for the presence of starch using an iodine solution. Then use a water bath to maintain the temperature. Then iodine solution test every 30 seconds. Then maintain pH using a buffer solution. Same volume of apple juice should be used. Same concentration of apple juice should be used. Same type of apple should be used. Same volume and concentration of enzyme. Repeat experiment 3 times and calculate mean. Turn the sharp side of the scalpel away from you when cutting the apple. But that's a safety procedure. So there's always, they expect you to give me these two you can write in every planning experiment. And you should write it because in order to get the 6 marks, the 2 marks, these 2 marks you can write in every planning question. So that ends this video on pectinase, and I hope this is going to be helpful for you in the upcoming exam.